Hello students, welcome to EPG Pathshala again. I am Dr. M.N. Gupta, Emeritus Professor from Department of Biochemical Engineering and Biotechnology at IIT Delhi. Today we are going to talk on the module Instruction versus Selection Theories from the paper on immunology. In the very first module on history of immunology, we had briefly mentioned the long-lasting controversies about the way antibodies with diverse specificities are created. Apart from its value as a historical perspective, a more detailed discussion about the basic philosophical differences between the instructive theories and selection theories is used. More techniques in biological sciences and biotechnology today use the concept of selection rather than instruction. So the objective of this module are to learn why and how instruction versus selection debate started. We will also learn the sort of ultimately how this debate gradually got settled. Of course, in this account, we will also understand first of all the basic difference between theories based upon instruction and selection. So the concept map of this module is very simple. We will once again delve deeper into the issue of generation of a specific antibody, because that is at the heart of the immune response. Then we will talk about the instruction theories, which were also dubbed under the title of the template theories. And we will see that on the other hand, there are bunch of theories which can be called the selection theories. We will also look at the issue of whether this generation of a specific antibody is the somatic, as perhaps the template theory would indicate, or is it genetic, as selection theories implies, because these two ways of generating specific antibodies would make us learn about the origin of the diversity of antibodies which the immune system is able to generate. The issue of instruction versus selection was crucial to the development of immunology. While chemists favored instruction theories, biologists believed in selection. Understanding this debate is important to the subsequent discussion in the later modules as well. To fully appreciate this debate, it is important that we also remember that the era in which this debate was taking place, and this encompassed a large number of years. It was not limited to an year or a month, but it lasted for a while. But what were the tools available for us and what we knew about the protein structure and function in general, because antibodies are proteins. And in fact, most of the antigens, experimental antigens or otherwise, are proteins in nature. Instructionist models focused on specificity of antibody and size of repertoire. The basic tenet of these models was that the information for the antibody to become a functional entity required the presence of antigens. Hence, these were also called template models, because the idea was that the antigen acts as a template on which the antibody molecule are created with the necessary specificity. The main proponents of these instructionist models were Brini and Horowitz around 1930, Alexander in 1932, Mudd around 1932, and lastly but not least, the Linus Pauling himself around 1940. In fact, Pauling, joining this way of thinking almost towards the end, gave fresh impetus to these instructionist model ideas. Selection models were based upon the belief that antibody repertoire pre-existed an antigen picked one somehow. The main proponents for selection models were Niels Jerne around 1955, Talmage 1957, and he again gave another version of his model around 1959. Burnett gave some ideas around 1957. Lederberg joined this debate around 1959. After Jerne has given some ideas, Burnett gave another version of his selection model around 1959. It is in the 1959 that Burnett outlined what has come to be known as clonal selection theory. He had also given an earlier version of the selection theory even before Jerne, but more on that later. These models were expected to explain the various critical features associated with the way immune system functioned. The first challenge was that any of these models must explain the capacity of the immune system to distinguish between self versus non-self. The second difficulty was to be able to explain presence of almost unlimited specific responses in terms of antibodies. That means no matter what antigen invades the system is able to create a specific response in terms of the antibodies. This is a very open-ended situation. The third, which was still very difficult for people to understand in those days, was the concept of the memory. Immune system seems to remember the previous encounter with the same antigen. Equally challenging was the possibility of being able to explain that how tolerance works. Tolerance not only distinguishes between self and non-self antigens, but having distinguished between the two, it tolerates self antigens and do not mount an immune response. The challenge was to have a theory which explained it all. The debate was also influenced by the stature of the proponents. Whether we accept it or not, the reality of the life is that the personalities unfortunately also matter in science. As we shall see, the debate was also shaped about how chemists and biologists tend to view science from their somewhat different perspectives. This is in fact an exciting story, and the best way, of course, to start a story is to go back to the beginning. It is necessary to understand how new disciplines start. In the beginning, what we call immunology was actually bacteriology. It started with Jerne and suddenly took a leap with Louis Pasteur. So the concerns were what causes disease, how to prevent them. Ironically, Louis Pasteur was not a medical person, nor was Elie Metchnikoff, who actually was an embryologist. We already referred that how Metchnikoff fiercely led scientist who thought that all immunity is cell mediated. Nevertheless, these two very different personalities kept the focus on pathogens and how our body deals with them. Pasteur's work raised the hope that all pathogens responsible for infectious diseases could be identified and vaccines for all produced. By around 1910, it was realized that it is not so simple. At this point, during the next 50 years, chemists became interested in how bodies fight diseases. Vaccinology morphed into a discipline which was more concerned with antibody-antigen relationships. It was during this period that Karl Landsteiner appeared on the scene. He was awarded the Nobel Prize in 1930 for the discovery of blood groups, which we know as part of ABO blood group system. Landsteiner's discovery of blood groups was a part of the larger set of studies. He can be considered among the top few immunochemists of his era. An important discovery by Landsteiner was the discovery of the concept of the hapten. As we have explained before, a small molecule which can trigger the immune response only when linked to a larger molecule. This included establishing that any compound arbitrarily picked from the shelf could provoke an immune response. So immune response was not just about pathogens and diseases, it was about antigenic compounds, antigenicity, which means the size and the structure of the foreign molecule. It brought chemists into play. Before biologists, it was actually chemists who related structure with a property. We tend to forget that the lock and key hypothesis for the enzyme action was given by Emil Fischer, who was a carbohydrate chemist venturing into the action of carbohydrases, glucosidases in his case. Landsteiner carried out extensive work on the structure and function relationship of antibodies, that is their specificities and equally important cross specificities. If you remember that all this was happening in 1940s, it was a powerful approach. This was also a time when biologists were looking at genes and trying to understand how they are produced. This claim that immune system could make an antibody tailored to any compound which need not have any biological relevance was at variance with that emerging picture. Landsteiner's discovery meant that the biological system recognized the structures which were not present in nature. Landsteiner's results in a way gave support template model, which was an instructive model or instruction model. As cells were unlikely to store information which will enable it to make a tailored antibody against an infinite number of heptane was difficult to understand. That is the difficulty our shop had in having an infinite stock. Landsteiner's heptane concept also meant that the shop had to also cater to a small pet which is accompanying a person. In 1940, Linus Pauling published a paper called A Theory of the Structure and Process of Formation of Antibodies in Journal of American Chemical Society. Pauling has been one of the greatest chemists. He is the one who had explained what a chemical bond is and given the valency bond theory. That is what we are now taught in school chemistry. All this is necessary to understand the impact of Pauling proposing a model for antibody preparation, antibody formation. What was known at that point? When an antigen is injected into an animal, some of its molecules are captured and held in the region of antibody formation. The number of binding sites on antibody was not known. Earlier, Merak, Heidelberg and others had suggested that precipitin reaction is a framework. Pauling agreed with that. Horowitz, Mudd and others suggested the idea of complementary structure for antibody and antigen. Ehrlich and Bordet implied it. So Pauling wrote, the rule of parsimony, that is, the use of minimum effort to achieve the result, suggests that there are only two such regions, that is, that the antibody molecules are at the most bivalent. While Horowitz in 1930 and Mudd in 1932 had suggested that antibody molecule had a different ordering of the amino acid residues, Pauling differed and said the difference is in the way the chain is coiled in the molecule. The point to note is that with so little to go on, Pauling was right about many fundamental aspects and so close to the real picture. It's interesting to note that Pauling acknowledged that his interest in immunology was awakened by conversations with Dr. Karl Landsteiner. 2 years later, Pauling published another paper called The Manufacture of Antibodies in Vitro using dyes like methyl blue and azophenyl arsenic acid and mixing these with normal serum globulin. He showed that serum globulin developed affinity for these compounds. These were taken as strong proof for the template theory or the instruction theory. According to Pauling's view, the compounds instructed the antibodies to be formed. Before we go further, it is necessary to explain that Pauling ran these experiments carefully and with proper controls. Unfortunately, what he did not know at that time was the non-specific binding which proteins display. Based upon these non-covalent interactions, macromolecules including proteins do show some imprinting effects under specific conditions. That Pauling observed these is curious. The template theory remained unchallenged most of the time. Its acceptance also arose from the fact that it was difficult to understand how otherwise an organism could contain such large information which would be required if a selection theory were to work. It may be added that there was another dimension of the debate. Was the origin of antibody diversity genetic in nature, or was it somatic in nature? Selection theories would suggest that this was entirely genetic in nature. Template theory suggested that antibody specificity and diversity was purely somatic in nature. What the template theory did not explain was the adaptive nature of the immune system. Immune system has a memory. Vaccinology initiated by Jerne and developed by Pasteur required it. Understandably, the challenge to the template theory came from the biologists. In 1995, Danish immunologist Niels Kaj Jerne recalled the earlier work of the year 1949 of two Australians, McFarlane Burnett and Frank Fenner. Burnett and Fenner had noted the following phenomenon, which template theory could not explain. Exponential rise in antibody production during the early stages. How do the antibody outnumber the template? It also could not explain the adaptive nature, as we have pointed out. As immune reaction progresses, antibody fits antigen better. This phenomenon is known as affinity maturation. Most important was the phenomenon of tolerance. Apart from referring to these concerns expressed by Burnett and Fenner, Jerne formulated a theory according to which diverse antibodies already existed. Once an antigen appeared on the scene, antigen-antibody complex was formed and it stimulated the white blood cells to produce the same antibody in larger amount. At the University of Colorado, Denver, David Talmage saw the link between Jerne's theory and an earlier theory by Ehrlich. Ehrlich decades back had proposed a side chain theory. According to that, white blood cells had side chains to which foreign substances linked chemically. This resulted in the cell producing large number of copies of that side chain, and excess side chains were shed in the blood. It is interesting to note that among other concerns, some scientists were worried about the chemical nature of the antigen binding to the side chains. Talmage linked the Ehrlich's ideas with Jerne's proposals. In 1957, he suggested cells are selected for multiplication, which match the invading antigen. He pointed out that as cancer cells produce homogeneous antibodies, cells can synthesize a specific antibody. Burnett had said that antigen triggers self-replication of cells. The nature of interaction with antigen was an issue. In later versions of his proposals, he felt that somehow antigen interacted with the genetic material of the cell. It is to be noted that it is in view of the vagueness on some aspects, selection theories could not match the clarity of Pauling's proposals. However, it is widely believed that Jerne and Talmage set Burnett on the right path. In his second version of his selection theory, he stated that the antibody pre-exists. Binding of an antigen with the cell receptor triggered the production of the same receptor. Each cell or its clone, that is offsprings, produced only one kind of receptor, side chain or antibody. This last feature was a leap of imagination. This also was the first time that clonal selection term was used. The term epitope was coined by Jerne. This still persists but is largely replaced by antigenic determinants. According to Jerne, an antigen has several epitopes. An antibody, apart from its own epitopes, has two paratopes. This term has fallen out of use now, but one may come across it in old textbooks and literature. The paratopes were essentially antigen binding sites on the antibody. Jerne clarified that paratopes are not epitopes but anti-epitopes, keys are keys and locks are locks. Jerne was referring to Fisher's metaphor for complementarity between enzymes and substrates. Audin, along with Maurice Michel in 1963, introduced the term idiotypy in French, which in English was written as idiotypy. Jerne and Janeway discussed Audin's concept of idiotypy, and Jerne defined idiotypes of an animal to be variable domains of the immunoglobulins present in that animal. The antibodies directed against idiotypes are called idiotypic antibodies. In 1969, Jerne visited Australia to attend Burnett's 17th birthday. The outcome was an article listing 10 things which were understood and 10 things which were not clear about antibody formation. In the second list was what controls the range of different antibodies that one animal or man can make. Jerne's idiotypy network theory thinks of the immune system as a network of mutually recognizing lymphocytes and antibodies. In this network, antibodies react with antibodies against them. Jerne basically thought that clonal selection theory thinks of a lymphocyte becoming competent upon exposure to the antigen. Jerne's view was that the immune system functions independent of a foreign antigen. According to his network theory, if the antibody was kind of a mirror image of an antigen in terms of complementarity of the shape, the antibody 2 against antibody would be similar to antigen 1. Thus, an antigen may fit some combining site among this interacting network. Thus, foreign molecules antigens merely modulate this network. The set of combining sites which recognize the epitope also anyway was recognizing another set of pre-existing idiotopes in the network which can be thought of as an internal image of the foreign epitope. Lymphocytes which display the internal image trigger duplication. Jerne's theory tried to explain all other features of the immune system. While Jerne's ideas received lot of support, the experimental data in support did not follow. Jerne was also awarded the Nobel for theories concerning the specificity in development and control of the immune system. We have learnt about Ehrlich's side chain theory, template theory, selection theories, idiotypy network theory, and finally the clonal selection theory. While clonal selection theory is now widely accepted, some of the ideas given by Jerne are still considered relevant. There has been a renewed interest in idiotypic interactions in the clinical context. Much later we will revisit that when we discuss autoimmune diseases. Right now, immediately in the next module, we will look at the clonal selection theory in more detail. Thank you.